Sa bawat kwento ng pag-unlad ay may mga institusyong nagsusulong nito. Institusyong nagbabahagi at tumutugon sa mga hamon ng panahon.
PCAARRD supports S&T Human Resource Development. Nagsusulong ito ng mga programa na naglalayong mapaunlad at mapayaman ang kakayahan ng mga researchers natin sa bansa at mga empleyadong katulad ko for better delivery of S&T products and services.
Sinusuportahan namin ang ating mga dalubhasa na kaisa natin upang umasenso ang ating bansa.
Tumutulong kami na maging makabuluhan ang mga teknolohiya upang magbigay ng karagdagang kabuhayan sa ating mga mamamayan.
Nagbibigay kami ng mga bagong kaalaman na teknolohiya na gagabay sa pagkalinga ng ating kalikasan at magpapaunlad sa kabuhayan ng mga magsasaka at mangingisda.
Isinusulong namin ang paggamit ng agham at teknolohiya bilang pagsagot sa mga hamon ng lipunan.
Kasama ninyo ang DOST-PCAARRD sa pagpapaunlad ng sektor ng agrikultura, aquatica at pangisdaan at likas na yaman. Sa gabay ng agham at teknolohiya, kami ay nakatuon sa pagpapataas ng produksyon ng pagkain, sa pagpapabuti ng kalidad ng pamumuhay lalo sa kanayunan, sa pag-aalaga at pagpapatatag ng ating kalikasan, at sa pagpapalakas ng kapasidad at pamamalakad sa pananaliksik at pag-unlad. Ang DOST-PCAARRD ay nangunguna sa pagbibigay ng kakayanan sa pagsasaka, aquatica at pangisdaan, at panglikas na yaman upang makipagsabayan ng sektor sa pandaigdigang pamantayan.
Narito po ang DOST-PCAARRD upang tulungan ang ating mga magsasaka, mangingisda at ang nasa sektor ng likas na yaman sa pamamagitan ng agham at teknolohiya. Isinasagawa ng DOST-PCAARRD ang aming mga programa sa pamamagitan ng pakikipag-ugnayan sa iba't ibang ahensya ng gobyerno, state universities and colleges at mga international partners. Ito ang DOST-PCAARRD. Kami ang DOST-PCAARRD.
The Institute of Crop Science is one of the academic units of the College of Agriculture and Food Science, UP Los Baños. The Institute is a merger of the former departments of Agronomy and Horticulture before it became Crop Science Cluster. In 2017, it started to be known as the Institute of Crop Science. Its mandates include instruction, research and technology development, extension and public service, and technical service. The Institute is composed of 5 divisions, namely Crop Biotechnology, Crop Breeding and Genetic Resources, Crop Physiology, Crop Production and Management, and Post-Harvest and Seed Sciences. The faculty, researchers, and staff of the Institute work together towards achieving a common goal to respond to the changes, challenges, and opportunities confronting Philippine agriculture in the 21st century. As an academic unit, iCrops offers 2 undergraduate majors under the BS Agriculture program of the College of Agriculture and Food Science. These are agronomy and horticulture major programs. iCrops also offers crop biotechnology major under the BS Agricultural Biotechnology program. BS Agriculture students are required to take foundation courses during their freshman and sophomore years. On their junior year, BSA students can apply under the agronomy or horticulture major programs and then specialize in crop physiology, crop production and management, landscaping, plant breeding, post-harvest science, and seed science and technology. Agronomy and horticulture major students can take several major courses. Since 2017, the Institute has produced 1 summa cum laude, 6 magna cum laude, and 32 cum laude. iCrops faculty members are the cream of the crop. Award-winning, outstanding young scientists, and UP scientists who embody honor and excellence. iCrops graduates are among the best in their fields and acquire top positions in research institutions, private companies, and government and policy-making institutions. iCrops graduates also become CEOs of their own companies and manager of their own successful farms. To add color to students' college life, iCrops is home to several dynamic student organizations. iCrops students are mentored to become the best that they can be, to be technically competent, skilled, and socially responsible while having fun and enjoying the journey.
We are iCrops!
Biotechnology has been an integral part to the further improvement of numerous sectors and industries, not just in the Philippines, but in the whole world. This is why yearly we gather to recognize, celebrate, and champion the technologies and people behind them that continue to work towards greater heights for biotechnology. I am Ross Penido, and this is the 19th National Biotechnology Week's webinar on SNT Agri-Aqua Biotech Applications. Isang maganda at makaagham na hapon sa ating lahat. Sa aming audience and speakers, magandang hapon po sa inyo. We can see that our live viewers are all excited about what's coming this wonderful afternoon. Hello to our friends from state universities and colleges, private universities, secondary schools, research and development institutions, farmers, and industry stakeholders. Thank you so much for being with us today. It is truly a sight to see that we are joined by 152 live viewers from our Facebook live stream alone. And to read some of the comments from our Facebook live stream, from Rochelle Azur Tumbaga, watching live from the College of Biliran Province State University, Biliran Campus. Hello po. Some more comments from Ryan Escote. Good afternoon. Watching from the Municipal Agriculture Office of Bulan, Sorsogon. Another comment from Ray Angelo Varela Salva. Good afternoon. Watching from the University of Southeastern Philippines. Welcome to this webinar, everyone. Do not forget to tag your friends in the comment section below or share this live stream with them so that they too will learn about the research initiatives featured this afternoon. Of course, follow us on our socials to be updated with our other initiatives. You may find DOST-PCAARRD on Facebook, YouTube, Twitter, and Instagram. Yan. I am sure that our viewers are excited for what's about to come. Our webinar this afternoon is simply star-studded with the Philippines' leading researchers and experts in the field of biotechnology. This is why I'm confident that a lot of you tuning in are eager to learn, so that I hope all of you will stay until the end of this webinar so that no one will miss out on learning from some of the Philippines' best and brightest in biotechnology. We will have an open forum later should you wish to ask for further information about our topics. Alright, I will not delay this any longer. To deliver the opening message on behalf of the DOST-PCAARRD Executive Director, Dr. Reynaldo V. Ebora, we have today the Director of the Crops Research Division of DOST-PCAARRD, Director Leilani D. Pellegrina. Director Lani, the virtual stage is yours.
A pleasant afternoon to our resource speakers, partners, farmers, students, organizers, and all our virtual participants. On behalf of our Executive Director, Dr. Reynaldo V. Ebora, the Philippine Council for Agriculture, Aquatic, and Natural Resources Research and Development of the Department of Science and Technology, or DOST-PCAARRD, is pleased to welcome you to this year's webinar on S&T Agri-Aqua Biotech applications. The Council annually conducts this activity in celebration of the National Biotechnology Week, or NBW, to feature some of our supported agricultural biotechnology-related research initiatives in collaboration with our partner agencies throughout the country. This event aims to showcase the results of years of hard work of the Council's supported initiatives in biotechnology applications and their potential contribution and benefits in agricultural sustainability. The NBW is held in observance of Presidential Proclamation Number 1414 Series of 2007, which institutionalizes its celebration every last week of November. This year, the Department of Agriculture spearheads the 19th NBW celebration with the theme Empowering Innovation for a Sustainable Future with Biotechnology in partnership with various government agencies, among which is DOST. The celebration highlights the role of biotechnology as a pivotal tool in achieving and sustaining food security, ensuring equitable access to health services, promoting a sustainable and safe environment, and driving industry development. Today's webinar will feature the accomplishments of 4 DOST and PCAARRD-funded initiatives on biotechnology implemented by the National Fisheries Research and Development Institute and the University of the Philippines, Las Bañas. Led by Dr. Mary Nia M. Santos of NFRDI, the applications of bacterial protein secretion systems in biotechnology will be discussed, focusing on how bacteria have developed sophisticated nanomachines. The talk will cover the role of Type 6 secretion system, or T6SS, during shrimp infection and the initial effort to explore its use for aquaculture biotechnology applications. From UPLB, Dr. Lilia M. Fernando Corpus will cover the portable nanobiosensor for fungal diseases of cacao, a nanobiosensor for the electrochemical detection of genomic DNA of Phytophthora palmivora in cacao. This completed project was able to validate and test a portable nanobiosensor technology for disease detection and eventual management of cacao disease. On the other hand, Dr. Eureka Teresa M. Ocampo will discuss the full genome sequencing of selected Philippine mango species, namely Wani, Paho, and Carabao, specifically Sweet Elena. The results of the project will be utilized in the marker-assisted selection and breeding as part of DOST-PCAARRD's mango breeding program. Last but not least, Mr. Mark Gabriel S. Sagarbaria will tackle the utilization of new breeding techniques for eggplant improvement, a project that generally aims to develop and deploy improved eggplant varieties with effective leaf hopper and eggplant fruit and shoot borer resistance derived from plant defense genes using innovative technologies. This is the first DOST-PCAARRD funded project that produced a gene-edited plant using the CRISPR-Cas9 technology. These biotechnology efforts are only among the many R&D initiatives of the Council. We hope that through this webinar, we will cultivate a deeper understanding and appreciation of the practical applications and benefits of biotechnology. I encourage the participants to have an engaging and relevant discussion during this webinar. I also hope that you will keep on supporting our activities such as this and rest assured that DOST-PCAARRD will continue to deliver more S&T innovations for the benefit of our fellow Filipinos in the agriculture, aquatic, and natural resources sector. Maraming salamat at magandang hapon po sa ating lahat.
Thank you, Director Pellegrina. It is truly a hopeful sight to behold that there are movements that continue to support and deliver S&T innovations, not only for progress, of course, but for the betterment of the Filipino people. Now, let us proceed and meet our first speaker. She is a BS Fisheries and MS Biology graduate from the University of the Philippines Visayas, or UPV. And she completed her PhD and postdoctoral training at the Academia Sinica in Taiwan. She has research experience in molecular biology, microbiology, biotechnology, and aquaculture at the Aquaculture Department of the Southeast Asian Fisheries Development Center at the Institute of Biology, UP Diliman, and Academia Sinica. He is a Fulbright Scholar and a Balik Scientist Awardee. Currently, she is the head of the Aquatic Animal Health Section of the Aquaculture Research and Development Division at the National Fisheries Research and Development Institute, or NFRDI. Introducing our first speaker, Dr. Mary Nia M. Santos, with her talk on the applications of bacterial protein secretion systems in biotechnology. Hello, Dr. Nia. Kamusta naman po kayo sa hapong ito? Alright, so it looks like may issue po tayo sa audio with Doc Nia.
Can you hear me now?
Yes po, ma'am. Yes po, Doc.
Yeah. Yes, I'm doing well. It's an honor to be here.
Thank you po, Dr. Santos. Para po sa kaalaman ng ating mga viewers, pwede po ba naming matanong kung ano po ang usual functions ng bacterial protein secretion system?
Okay, so I'll discuss it sa talk, pero mainly, meron siyang two functions. Unang function niya, antibacterial weapon siya. So, ginagamit siya ng bacteria para mag-survive and thrive sa environment. Yung isang function niya for virulence or for pathogenesis dun sa host like sa plants or sa fish.
Yun. So, all right, ma'am. Thank you so much for that answer, Dr. Santos. I believe our audience is ready. Dr. Santos, please take it away.
Good afternoon, everyone. It is my pleasure to share with you the applications of bacterial protein secretion systems in agriculture biotechnology. So bacteria has evolved this nanomachine called protein secretion system to secrete proteins or virulence factors to another host cell or into the host environment. So far, there are already 11 secretion systems that has been characterized. And these nanomachines are used for pathogenesis and survival. So as an example of pathogenesis, so several type protein secretion systems like type 3, type 4, type 2 are used by plant pathogens. While the Vibrio fischeri, symbiont of the bobtail squid, uses type 6 secretion system to establish its symbiosis in the light organ. So as you can see here, only Vibrio fischeri strains that have the type 6 secretion system can survive in the light organ. So to my knowledge, this is the most advanced application of this nanomachine. And in this group from Switzerland, they were able to successfully deliver bacterial, viral, eukaryotic proteins that are fused to type 3 secretion system substrate, as shown here, YUP-E, of different porous sizes of substrates and deliver into wide range of target cells, and it is in a fast and controllable manner. So compared to other conventional methods, this novel technology has several advantages. So it is extremely powerful and efficient. The protein can be delivered within a minute, and multiple proteins can be translocated simultaneously. And this from basic research, so now this is already at the phase 1 of clinical trial. And this young researchers established a startup company called T3 Pharmaceuticals. And they are developing a bacterial cancer therapy platform using type 3 secretion system. So they use this machine to deliver therapeutic proteins to activate immune cells, which in turn would result to the regression of primary tumor and metastasis. So in this talk, I'll be focusing on type 6 secretion system. So it is a bacterial nanosphere gun. So if it's like a gun, it also has a bullet, which is called an effector or toxin. And it works like a power drill as shown in this video. And as you can see here, this is the machine inside the bacterial cell. So it is present in about 35% sequence of gram-negative bacteria and all encoded by human, animal, plant pathogens, and even non-pathogens. So their known functions is for survival. It increases the fitness and survival in the environment or in the host, and it can also be used for pathogenicity and acquisition of metal ions. In agriculture, this type 6 secretion system can be used as a biocontrol agent against plant pathogens. So Pseudomonas butida has been labeled as a plant warden. In this study, they applied Pseudomonas butida and Xanthomonas campestris, and the results showed that there is a reduced necrosis on the leaves, and this effect is dependent on the type 6 secretion system activity. So, and recently, this review has just been published this month on how type 6 activity can be harnessed into different biocontrol strategies. First is the production of antagonistic bacterial or fungal effectors to target plant pathogens and suppress them. Second is for the metal uptake activity. So we could exclude pathogenic microbes by depriving the uptake of metal ions or essential nutrients to further suppress their growth and viability. And then the response to environmental cues. We could take advantage of this for developing tunable expression of the machine. We could turn it on or off based on the environmental cues. And then next is the DNA uptake. So we could also develop type 6 to facilitate the acquisition of genes that confer niche adaptation or enhance its survival in the environment. So this is a new development in the field of type 6 secretion system. So this group has generated strains expressing surface displayed antibodies called nanobody, which is directed at unique surface antigen of the target cell. So this is used for selectively targeting bacterial cells in a polymicrobial community. And so now the PI, the principal investigator, is now in Academia Sinica in Taiwan, and we are collaborating with him and his group for the newly approved DOST project next year. Another perspective is that we could also use a community or consortia applications of biocontrol. We could combine strains with different modes of action for biocontrol activities, such as creating blends of different toxic metabolites. Another exciting development in the field is the use of type 6 secretion system to deliver exogenous effector and genetic editing protein into target bacteria. So we could integrate this machine in the CRISPR-Cas9 system. So this study shows the potential of the type 6 mediated delivery in genome editing, killing drug-resistant pathogens, and delivery of protein-based drugs and antigens. So I'll be sharing the ongoing project on the role of type 6 secretion system during shrimp infection of Vibrio-causing append. And this is our initial effort to use the type 6 secretion system for the disease management of shrimp. So this is under the Biotech program, and we are cooperating with Academia Sinica in Taiwan. So in this project, we are using Vibrio-causing append as chassis strain for future applications, like for prevention, prophylactic strategy, like developing antigen delivery system. This is for disease control, to develop next generation microbial, or for treatment, or using the type 6 secretion machine as a delivery system. So this better understanding of the molecular mechanism of this system in aquatic pathogens can help develop more targeted strategies or therapies for the aquaculture industry. So what's the difference between append and non-append causing Vibrio parahemolyticus? So for append causing Vibrio parahemolyticus, they just have this extra chromosomal plasmid which encodes PRAB toxins. However, recently, Young et al. Found that several append causing Vibrio parahemolyticus does not have this toxin. So there might be other virulence factors that are causing. In a separate study, in a comparative analysis of Vibrio parahemolyticus, this group has found that only append causing Vibrio parahemolyticus encodes the type 6 secretion system. And now it's not only Vibrio parahemolyticus that's causing this disease in shrimp, but other bacteria such as Vibrio harveyi, ONC, Campbelli, and Pinensis. So we have 3 strategies to address the objectives of our study. Base is the comparative, first is the comparative genetics. So we did the whole genome sequencing and assembly of append and non-append causing Vibrio parahemolyticus and Campbelli. So we found that in Vibrio parahemolyticus PH1339, which is the append causing strain, it encodes both the type 6(1) and 2 clusters. And while the non-append causing only includes the T6SS2. We also did a phylogenetic analysis for both strains and analyzed the associated differentiation of type 6 and appen. So we found that majority of the strain encode the T6SS1 and our appen strains, but we did not find any strict correlation between the presence of T6SS1 and appen. So now we are looking at the specific regions in the effectors, which could confer the specificity in appen causing strains. Next is the functional analysis. So we have found that Vibrio parahemolyticus PH1339 has toxic activity against bacteria. So to know which is the factor that's causing this toxic activity, through our bioinformatic analysis, we found 4 putative effectors. And we expressed this each of these putative effectors and the growth inhibition assay results that one of them may be responsible for the toxic activity, as you can see here in the decrease in the growth inhibition when we express the TSE3 effector. And lastly, for the ecological relevance, so we did challenge assay for the animal study. So we infected Penaeus baname shrimp and found that Vibrio campbelli PH1401, which is an appen causing strain, results in about 87% mortality. So we are continuing with this project, and we really hope that we could translate the research results of this project into something useful for the industry. So far, this is the accomplishments of the project. We have submitted the whole genome sequence in GeneBank and published the genome of Vibrio parahemolyticus PH1273 in microbiology resource announcement. We have presented the initial results of our study in a conference in ASM Microbe 2023 in Texas. And we have also capacity building for our staff and also for the students from UST and PLM. And we have partnerships with Academia Sinica, CIFTEC, and B4 NFLT. So yeah, thank you. So if you have any questions or clarifications, you are welcome to ask. And also, we will have a new project next year on the development of probiotic bacterium expressing nanobody and or dsRNA against shrimp viral pathogen white spot syndrome virus. So if you're interested to join the group, so just feel free to contact me. Thank you.
Thank you, Dr. Santos. It is quite a curious and remarkable sight to see that from small organisms like bacteria, people like Dr. Santos and her team are able to leverage their natural yet sophisticated functions to explore options for enhancing aquaculture. Through these applications of biotechnology, we can see absolutely limitless opportunities to improve the livelihood, food quality, and resources of Filipinos. Once again, thank you po, Dr. Santos. A reminder pala to our dear audience, after our second speaker, we will be having our first open forum for today's webinar. So I hope na ready po ang ating mga questions for our first two speakers. Don't miss out on asking some of our leading experts on biotechnology. Now, let's keep going, shall we? Introducing our second speaker. She is a biochemist whose research is on the utilization of secondary metabolites from plant growth-promoting bacteria for plant production and propagation. She works on the synthesis, characterization, and utilization of biocompatible nanomaterials for the improvement of crop productivity, along with safety studies of nanomaterials in plants and microorganisms. Her research team also develops nanobiosensors for various applications. She is an associate professor at the Institute of Crop Science and an affiliate professor Phil Professor at the Institute of Chemistry, University of the Philippines, Los Baños or UPLB, where she teaches agricultural biotechnology courses and biochemistry courses. She is the current Associate Dean for Research and Extension of the College of Agriculture and Food Science, UPLB. She is the co-founder of Makiling Tech Inc., a university spin-off which aims to facilitate the commercialization of microbial inoculants and nanobiotechnologies developed by their team through the Department of Science and Technology and other granting agencies. Her BS in Chemistry, MS in Biochemistry, and PhD in Biochemistry were from the University of the Philippines, Los Baños. She took her post-studies at the Massachusetts Institute of Technology. Here to discuss the portable nanobiosensor for fungal diseases of cacao, introducing Dr. Lilia M. Fernando Corpus. Good afternoon po, Dr. Fernando Corpus. Kamusta po kayo?
Good afternoon, Ross. Mabuti naman.
We're very glad to hear that, Doc. I'm sure this is a point of curiosity for our audience po. Dr. Fernando Corpus, can you please briefly explain kung ano po ang nanobiosensors?
Okay, so nanobiosensors, from the name, for detection ito ng biological materials, can be the whole microorganism or part of that organism like DNA or protein, and meron tayong nano. So gagamit tayo ng nanomaterials.
All right, thank you for that answer, Dr. Fernando Corpus. Let's proceed to our second topic. Dr. Fernando Corpus, the virtual stage is yours.
Good afternoon, everyone. Before I start, I would like to thank the DOST-PCAARRD for allowing us to share our work on portable nanobiosensors for the detection of pathogens in cacao. Nanotechnology has gained intense attention in the recent years due to its wide applications in medicine, agriculture, and the industry. The utilization of nanomaterials has been used in biosensors to enhance their efficiency and sensitivity. These are some of the common nanomaterials used in medicine, agriculture, and the industry. For nanobiosensors, our team used the ferric oxide nanoparticles or the magnetic nanoparticles and the gold nanoparticles. Our nanobiosensor technology started through our collaboration with the DOST Balik scientist, Dr. Vanji Alusilha. She is from Michigan State University. So we adapted at first her prototype, wherein you have the analyte. This can either be a DNA or an antigen from the target pathogen. A bioreceptor can be an oligonucleotide probe or an antibody. A transducer, which can also be a nanoparticle, a signal amplifier, and detection, which can be through electrochemical or colorimetry. For electrochemical method, later on, I'll be showing you our biosensor platform, which is the screen-printed carbon electrode. Some advantages of nanobiosensors over conventional methods of detection are rapid detection time, high sensitivity and specificity, and compatibility with data processing technologies. Compared to the common methods of detection of plant pathogens like that of ELISA and nucleic acid-based techniques like PCR, nanotech-based detection have higher specificity and sensitivity. These are some of the nanobiosensors that we've developed through the years in collaboration with Michigan State and through support from the DOST-PCAARRD, the DOST-PCAARRD, PCAARRD, and the Grand Challenges Canada. So for the detection of food and waterborne pathogens, we have a prototype for E. coli, Salmonella, and Listeria detection. For plant pathogen detection, we have that for cacao, which I'll be sharing later on. This is the prototype for the electrochemical detection. And lastly, we have the detection of viruses, okay, from mosquitoes, specifically that of dengue and JEV. Our work on nanobiosensors has the following program. So we have that for the food and waterborne pathogens, plant diseases, tropical diseases, and we're focusing on the detection of these viruses in mosquito. We also hope to collaborate with the animal science and veterinary science for bacterial and chemical contaminants detection, inorganic fertilizer for the detection of salmonella, and to collaborate eventually with the Department of Health for the detection of bioterrorism agents. So now we'll focus on the detection of diseases in plants. The FAO estimates that annually between 20 to 40% of global crop production are lost due to plant pests. And imagine there's a loss of about $220 billion in the global economy due to invasive insects. With increased trade and travel, the risk of plant pests spreading into new areas across borders is now higher than ever before. We witness shocking number of threats to the well-being of our plants. I remember nung meron tayo nung coco lisap, and thankfully we were able to move on and have some control for this insect pest in our coconut industry. So these threats in plants have to be addressed. And by extension, kasi maapektuhan din po eventually ang ating health, environment, and economy. We have to be very proactive in terms of biosurveillance, especially of emerging plant pests or pathogens. So as an example, our project in cacao focused on the detection of the causal pathogen on one of the diseases, which is the black pod rot, which accounts for up to 70 to 80% loss in cacao production. One of our research associate and also became our master's student, Mr. Anthony James Franco, worked on a DNA-based electrochemical nanobiosensor for the detection of Phytophthora palmivora. The common methods of detection are culture, PCR, and ELISA. And usually these conventional methods are laborious, requires complicated instrumentation and skill level of personnel. And we want that the detection be done in the field, on-site detection po tayo with the farmers, and we want to detect the causal pathogen before the onset of symptoms in plants. So these are some images of infected cacao pods with Phytophthora palmivora and the culture that we've isolated from the infected samples. So the biosensor technology starts with the triage technique. We call this the triage technique. So overall, our detection makes use of magnetic nanoparticles and gold nanoparticles. So the magnetic nanoparticle is used in the screening or the triage technique. So imagine you have a lot of samples from the field. You have to check first or screen which among these samples have pathogens. So the triage technique shows a matting or spreading of the magnetic nanoparticles if it's positive for a pathogen. So only those that are positive from the triage technique will be used for the DNA-based detection using gold nanoparticles. So our dream is to have a biosurveillance system that when we were able to detect a pathogen in a particular location, we can send it to a database or a cloud wherein our regulatory agencies will be able to see this and then be able to work on biocontrol or other approaches in treating yung soil or yung plants mismo natin. that are infected with pathogens. So for the screening technique or the triage technique, this was first used by the team of Dr. Alusilha in dengue detection. So when she visited for, I think, the third time the country, she asked us to explore the application of this screening technique in plant pathogens. And fortunately, we were able to detect Phytophthora palmivora in the matting technique that she introduced to us. So from the matting technique, we developed a DNA-based biosensor, and starting with electrochemical detection, we used this prototype. So this is a handheld potentiostat, and this is battery operated. So we have a sample to be prepared. So for instance, we're in the field, we extract the DNA on site, and about a drop, about 50 microliters or 100 microliters of the prepared sample is just placed in this circle here in the screen printed carbon electrode. So overall, the sample preparation may take about 3 to 2 hours. We're still optimizing yung part po na ito sa field detection and extraction of the DNA, but the detection using the electrochemical method is about 1 minute lang po. It's the sample preparation that takes time. But compared to ELISA, ELISA is antibody-based or protein-based, and PCR is DNA-based. So yung culture method naman, it will take probably more than a week before we can confirm the presence of a pathogen. Okay, so just to show you how DNA-based detection works. So in this example or illustration, we have PANI, which is the magnetic nanoparticle. Ito pong gray circle. And this is oligoprobe 1, yung parang tail ng magnetic nanoparticle. And we also prepared another oligoprobe, which we call the capture probe, different from that of the detector probe here. And it's attached to biotin. Later on, we'll know why it's important for it to be attached to biotin. So if we have the target DNA, for instance, for Phytophthora palmivora, so it will hybridize with our oligoprobes that were designed. And because the oligoprobes are very specific to a specific sequence in the target DNA of the pathogen. So in the field, we don't make use of centrifuge or other instrumentations. By virtue of the magnetic property of the nanoparticle, we can purify the sample through magnetic separation. So once we have the purified sample, we use the screen-printed carbon electrode embedded with gold nanoparticle, the yellow circles here to amplify signal. Tapos yung isa po ay streptavidin. So streptavidin and biotin have very special affinity. So yan po yung magka-capture dito sa ating platform nung ating purified sample with the nanoparticles and the target pathogens DNA hybridized to our 2 oligoprobes. So from here, we can perform electrochemical detection using the battery-operated potentiostat. So here's an example of the electrochemical detection. So in the field, meron po tayong scale para madaling makita ng farmer or any regulatory officer using this method kung positive or negative. So we just did some laboratory scale optimization to confirm yung detection po. So for this voltammogram, this is the blank, yun pong pinakamanipis dito na area, and the other voltammograms here have bigger area corresponding to the increased current when the pathogen is present. So we compared the selectivity of the designed oligoprobe with that of other plant pathogens. And for Phytophthora palmivora, we found that it's very selective, yung ating pong oligoprobe to our target pathogen. And for the limit of detection, we can detect for as low as 0.1 nanogram per microliter of the DNA. So again, this is our prototype for the electrochemical detection of the Phytophthora palmivora. Now we have another project on the validation and field testing of the portable nanobiosensor. So this was done by one of our research associates, and also she had this as her master's thesis, Miss Marinold Purificacion, and she focused on the detection of the causative pathogen for vascular streak disease, which is Lashodiplodea theobromei. So this is our nanobiosensor kit. So for the screening technique, still she used the magnetic cell capture or the triage matting technique. So how is this done in the field? So you have a buffer in a tube, and you have here your sample from the plant. Then you just add the plant sample to the buffer, add magnetic nanoparticle, mix and incubate at room temp. You separate the contaminants from your sample through magnetic separation. Okay, so eventually you observe yung matting po ng magnetic nanoparticle. So same as earlier, yung spreading ng magnetic nanoparticle indicates the presence ng pathogen. But not specific yet, it's just possible that this has a pathogen. So it's in the DNA-based detection that we confirm if our target pathogen is really there. Okay, so we're working on quantification ng ating pong triage technique. So it should have a higher mean threshold intensity pag meron siya nung target pathogen, pag meron nung matting compared to the blank. So for the DNA-based detection, now I'll give you an example of our colorimetric method for Lashodiplodea theobromei. So we have here the red circles as the gold nanoparticles, and ito pong nasa surface niya are functionalized oligoprobes specific to Lashodiplodea. So if you have the target pathogen, you will retain the pink solution. Pag wala po nung pathogen na target natin, nag-aggregate yung gold nanoparticles, so it results to a purple to clear solution with precipitate. So this is another illustration. So pag positive siya sa Lashodiplodea, walang change in color, it's still pink. But without, pag negative siya dun sa Lashodiplodea, pink to purple with a precipitate of the gold nanoparticles. So these are some pictures obtained from actual detection of Lashodiplodea theobromei. So the blank is purple and pinkish po itong Lashodiplodea. Another illustration, so we have clear with, di po kita yung precipitate dito. So the oligoprobe is specific to Lashodiplodea theobromei. That's why we're not able to detect Phytophthora palmivora here. But even if you have them both in your samples, you can still detect yung Lashodiplodea. So we have a lower absorbance for Lashodiplodea because it retained yung kanyang pink solution at 520 nanometers. Other applications of nanobiosensors is for the detection of other pathogens in banana, tomato, potato, peanut, mung bean, and other crops. So we use the same technique, the triage technique and DNA-based detection. So some examples of triage technique used in the detection of BBTV or the banana bunchy top virus, Fusarium TR1. In here, we see the matting compared to the blank. Ralstonia solanacearum. So we have matting here for positive detection of Ralstonia. And for the DNA-based detection, so we have Ralstonia detected in these pink solutions on the left. And we also have a project that's ongoing on the detection of mycotoxin-producing organisms using nanobiosensor. This is being done by Miss Kimberly Suministrado. She is a research specialist and also a master student doing her thesis on this project. So we want to lessen yung losses po natin sa post-harvest problems on cacao beans due to yung mga reject, yung presence po ng mga biological contaminants. So Aspergillus species can produce aflatoxin and ochratoxin, and these are detrimental to our health as consumers. So applying the same method, the cell capture or the triage technique using magnetic nanoparticle and eventually DNA detection is also applied in this technology. So this is the triage technique for Aspergillus species. And for the colorimetric, these are some optimizations that were done on several parameters for the detection of the two Aspergillus species producing aflatoxin and ochratoxin. So the team also conducted trainings and seminars with farmers and cooperators in Davao City on how to use the portable nanobiosensors, and also with farmers from Benguet. And we are also encouraging students, promoting the use of nanobiosensors for them to learn more on this nanotech innovation through our anniversary week and exhibits in the university. So this is our alert nanobiosensor, our prototype. So we already applied for IP protection, for patent and trademark for the technology. So we're still working on some validations, field validations for the developed nanobiosensors. So hopefully we can market the technology soon to be used by our farmers and our regulatory agencies. So our team would like to address SDGs 3 and 12, which are good health and well-being and responsible consumption and production. On behalf of our team, thank you and have a pleasant day.
Thank you so much, Dr. Fernando Corpus. We can all be sure that the technologies developed by Dr. Fernando Corpus and her research team are here to make changes that will certainly stay. Through works like this, we are given a glimpse of the rewards of the union between chemistry and biotechnology. We are also reminded that biotechnology is applicable, promising, and effective in various disciplines and industries. Once again, Dr. Nando Corpus, thank you. We are certainly on a roll. With that said, I hope ready na po ang ating questions for our first two speakers. You may send your questions by typing in the comments section of our FB live stream. Once more, we would like to call on our first two speakers, Dr. Santos and Dr. Fernando Corpus, to the stage. Hello po ulit.
Hello Ross.
I hope ready na po tayo to answer our audience questions.
Yes.
Yan. Mukhang ready na po ang ating first two speakers. So, allow me to read. This is from John Paul Ballesteros of Malolos, Bulacan. What are the other biocontrol applications of T6SS? So, I think this question is directed to Dr. Santos.
Yeah, thank you for that question. So, tulad ng na-discuss ko kanina, there are many biocontrol applications for type 6 secretion systems. So, we can use it actually as a next generation antimicrobial. So, we could use it to kill pathogenic bacteria, yung mga disease causing na bacteria dun sa aquaculture systems. Or pwede rin i-suppress yung growth nila using this system by using yung function ng type 6, like yung pag-limit ng nutrients. Like, for example, we could engineer a bacteria na nagsisequester ng mga iron or other nutrients in the environment. So it could limit yung growth ng mga pathogenic bacteria sa environment.
Yan. Thank you po, Dr. Santos. Now, for another question, please. This is from Sorjun. How extensive is the adoption of the typical farmers to nanobiotechnology? There are lots of potential microbial symbiotes in the soil. Aside from bio and oikova, are there more of these? So I think this question is directed to Dr. Fernando Corpus.
Okay, thank you, Ross. With regards to BioN and Mycobam, these are actually biofertilizers or microbial inoculants. Wala pa sila nung nanomaterials, no? In terms of adoption, alam namin yung mga farmers gustong-gustong gamitin, no? Focusing on BioN and Mycobam muna. But of course, we have a guideline in the university in terms of tech transfer. But for BioN, I think BioN is already transferred, no, to an industry partner. So, hindi na sa UPLB ang production natin for that. And unti-unti naman, lumalabas na ng university yung ating mga technologies. For nanobiotech products, we have to comply with the need not to register as well sa Fertilizer and Pesticide Authority. Same guidelines being done with the biofertilizers and other inputs, no, tulad ng inorganic fertilizers din. So, for the adoption, meron kasi tayo diyang parang stages pa before we go into adoption. Parang sa nag-a-adapt ka ng mga bata. So, may mga tech transfer mechanisms tayo for that. But the university, through the facilitation of DOST, specifically PCAARRD then for our technologies, marami ng mechanisms para mapabilis yung commercialization ng mga developed products. And when we go to trainings and extension activities, very receptive naman yung mga farmers natin. Ang laging tanong nga nila, kailan po namin ito pwedeng mabili na nandito na sa aming tabi? So, very naman po, happy sila lalo na when you say that these are Philippine-made. Talagang inaantay nila. So, papunta na talaga tayo sa route na hopefully, no, maging sustainable din yung ating country na sana yung mga gantong technologies hindi na natin i-import. Meron na nung sariling atin. And there are industry partners in the country na willing to manufacture in larger scale yung ating mga nanobiotechnologies or even yung through classical biotech na na-develop natin. Back to you, Ross.
Thank you for that answer, Dr. Nando Corpus. That's very encouraging. Now, again, next question, please. This is from Ernie Patricio of Cebu City. What are the practical applications of bacterial protein secretion systems, especially in the aquatic sector? I think this question is again directed to Dr. Santos.
Yes, thank you for the question, Ernie Patricio. Okay, so dalawang naisip kong practical application niya. No? First, yung ginagawa na namin ngayon on disease management. And second, dahil sa development dun sa type 6 secretion field, it could also be used sa breeding. So dun sa disease management, we can use it like in 3 ways. First is yung prevention. So we could develop the type 6 as antigen delivery system. Second, pwedeng yung biocontrol, which I also explained in the presentation. Pwede tayong gumawa ng mga next generation antimicrobial using this machine. And third, pwede siyang gamitin din sa treatment. So instead na yung mga toxins or harmful proteins, yung bullets or sinushoot up niya ng machine, we could use like therapeutic proteins or bioactive compounds yung gagamitin na bullet ni type 6 secretion system to deliver dun sa target cells, either sa bacteria or sa eukaryotic host niya. So, yeah. Thank you.
Thank you so much for that answer, Doc Santos. Now, next question, please. This is from Albert Espino of Caloocan. What is the advantage of a DNA-based nanobiosensor over protein-based sensors? So I guess this is directed once again to Dr. Fernando Corpus.
Okay, thank you, Ross. So for a DNA-based nanobiosensor, you remember during the COVID-19 pandemic, meron tayong yung rat, tsaka yung PCR-based. So, yung antigen-based, pwede rin yun yung protein-based natin because basically, antibody and antigen or antibodies are proteins. So, hindi siya ganun ka-specific pa, no? Kasi yung antibody lang yung dinidetect niya. When we go to the more specific na DNA-based, so that's when we're able to at least ma-narrow down natin na yung organism na yun talaga yung ating dinidetect. So it's more on specificity, yung difference ng DNA-based kaya ng protein or antibody-based detection. And another thing na mahalaga din, usually ang antibodies temperature sensitive. So medyo kino-consider natin yon when we use it in detection. At least for DNA, somehow stable siya no for certain temperature room or pwede rin no yung mga field temperature natin. Thank you.
Thank you po, Dr. Fernando Corpus. Another question, please. This is from Lynn B. Resurrection. Besides cacao, was the nanobiosensor already used in other crops besides cacao? What are the results? Thanks po. I guess this is directed po to Dr. Fernando Corpus.
Okay, thank you. So I mentioned earlier, we also tried using the nanobiosensor in banana bunch-top virus detection, Ralstonia, which is a pathogen in tomato, sa Fusarium, no? Pero TR1 lang po, hindi kami nakapunta noon sa Davao. Alam natin yung Fusarium is a problem sa ating banana industry up to now. Actually, sabi na nga ng mga farmers sa Davao, even the companies there, hindi na kailangan ng detection treatment na. So baka may mga colleagues tayo who can help sa Fusarium treatment sa Davao. And also in mung bean, peanut, and white potato. Ayun po. Tapos na kami dun sa laboratory scale, development ng prototype, nasa na kami, validation stage for the other crops.
Thank you po, Dr. Nando Corpus. Following question, please. From Janelle Andal of San Pablo, Laguna, what is the function of T6SS? This question is directed to Dr. Santos.
Yeah, yung function ng T6SS is three functions mainly. So for antibacterial siya. So ginagamit siya for the bacteria to survive dun sa environment niya. So it was found na if yung bacteria nag-encode ng T6SS, meron siyang competitive advantage over dun sa other bacteria na hindi nag-encode ng machine na ito. So, ibig sabihin, mas nagtatrive sila, nagsasurvive sila sa environment because of this machine. And then second, yun, ginagamit nila yung mga effectors or toxins para i-manipulate yung host o kaya mag-thrive din sila sa environment. And then thirdly, ginagamit din for acquisition ng metal ions. Yeah. Back to you, Ross.
Thank you po, Dr. Santos. Now, the following question, please. This is from Rogelio Espinosa Magbitang. In detection, is it possible to detect the pathogen in a deceased plant? Or would it require to be isolated and purified first? So this is directed to Dr. Fernando Corpus.
Okay. So, yes, we can also detect the pathogen dun sa plant na talagang kita na yung symptoms. And as I mentioned, we can also detect this in the soil bago tayo magtanim. We just need to isolate yung DNA para dun sa specific detection. And that's tricky because we want this to be on-site. So we already have an extraction technique that can be done sa field without siyempre tayo gagamit ng sophisticated equipment. So yes, we can do detection. No, we need to isolate the pathogen or the DNA first na nandun sa ating infected plant para malaman natin kung anong pathogen yun. Kasi doon talaga natin malalaman kung anong treatment or biological approaches, chemical approaches yung gagawin natin no, para makontrol yung pagkalat ng pathogen. Ayun po.
Thank you, Dr. Fernando Corpus. For the last question, this is from Kimberly Bernabe of Lumban, Laguna. What are some challenges in the development of a DNA-based nanobiosensor? This is for Dr. Fernando Corpus.
So, yun, kakatapos nung isang question kanina, yung DNA extraction. Mahirap siyang gawin, yung on-site, no? So, pero ano naman na, promising yung technique namin ngayon na may gagamitin ka lang piece of paper, then you can extract the DNA already. But we're still optimizing some of the procedures here. So, yun po, yung sample preparation ang challenge dito. Okay na po.
Thank you so much. Thank you so much po, Dr. Fernando Corpus. Thank you so much, Dr. Santos and Dr. Fernando Corpus. Your insights and expertise are well appreciated. With that, natapos na po ang ating first open forum. Meron pa po, so stay tuned po sa ating audience. If any of our viewers today have further questions, you can reach us through our email at crd@picard.dost.gov.ph. You can see the same email address flashing on your screens. It's below right here. And as we close our first batch of speakers, we would like to award the certificates of appreciation to Dr. Santos and Dr. Fernando Corpus. Please allow me to read the citation. The Department of Science and Technology, Philippine Council for Agriculture, Aquatic, and Natural Resources Research and Development, or DOST-PCAARRD, and the University of the Philippines Los Baños, or UPLB, present this certificate of appreciation to Dr. Marinia M. Santos for serving as a resource person for the webinar titled SNT Agri-Aqua Biotech Applications, held on November 21, 2023, 2 p.m. via StreamYard and Facebook Live, given this 21st day of November 2023. Signed by Dr. Reynaldo V. Ebora, Executive Director of DOST-PCAARRD, and Dr. Rainiel S. Pamplona, Project Leader from UPLB. Thank you po, Dr. Santos.
Thank you.
Thank you po. And we would also like to give the same certificate of appreciation to Dr. Lilia M. Fernando Corpus. Again, thank you very much po for sharing your knowledge with us this afternoon. Maraming salamat po, Doc Nia and Doc Lia.
Thank you.
Thank you po. Maraming salamat po ulit. Without further delays, let's meet our third speaker for the day. She is a researcher who has led projects on screening for drought tolerance in legumes, where she unraveled mechanisms for drought resistance in mung bean. As a plant physiologist, she collaborated with maize breeders on the development of double stress tolerant maize varieties and the population development of native corn for different environmental stresses. She was also the co-developer of the immensely popular SNAP Hydroponics, a low-cost system for hydroponic culture of high-value crops and the complement SNAP nutrients. One of her studies was the first to report on a specific transcription factor gene for ripening control in bananas. As a professor, she has advised more than 50 students under the BS Agriculture, BS Agricultural Biotechnology, MS Molecular Biology and Biotechnology, and MS and PhD in Agronomy and Horticulture degree programs. Currently, she is a Professor 1 and the Director at the Institute of Crop Science, or ICROPS, College of Agriculture and Food Science at the University of the Philippines, Los Baños. Everyone, introducing Dr. Eureka Teresa Ocampo for her talk on the full genome sequencing of selected Philippine mango species. Good afternoon po, Doc Weng. How are you doing this afternoon?
Hi, Ross. Okay naman ako.
Glad to hear that po, Doc. Para naman po sa ating live viewers, can you tell us what genome sequencing is?
Okay, to put it simply, it's genome sequencing is just taking all the DNA in one organism, and then knowing the sequences or the sequence of the different bases that are in the DNA. So yung lahat-lahat ng DNA, so we call it genome. And then pag sunod-sunodin lang natin yun. So that's
Thank you so much po. All right. Thank you so much po, Doc Weng. I think ready na po ang ating viewers for your talk. Doc, please take the virtual stage.
Today, I will be presenting the full genome sequencing of selected Philippine mango species. This is our work that was implemented at the Institute of Crop Science here at the College of Agriculture and Food Science, UPLB. So the source of funds was DOST-PCAARRD, GIA, for funds totaled 7,799,208 pesos. And this was implemented from June 1, 2020 to August 31, 2023. So it was a full 3.5 years. So mango, our favorite fruit, it's fleshy with varying sweetness and textures depending upon the cultivar. So the national fruit of the Philippines, and it is also the third top fruit export after, of course, banana and pineapple. The Carabao mango is the main export variety of the Philippines and is known worldwide for its superior fruit quality, exotic taste, and aroma. And the Philippines has a large germplasm of mango genotypes that are maintained primarily in Los Baños and in Guimaras, Iloilo. So based on the Philippine Mango Roadmap for 2021 to 2025, the Philippine mango industry has seen a decline in the production in the past 5 years. So the volume has declined, the expansion of areas has remained stagnant, and so are the number of trees that are harvested, declining yield per hectare and stagnant yield per bearing tree, declining mango exports, increasing production cost, but decreasing farm gate price. So this is the DOST-PCAARRD mango breeding program, where our team has been involved with. So we are involved with the program on enhancing competitiveness of Philippine Carabao mango through varietal improvement program. So here we were involved in marker-assisted breeding for mango. And of course, the full genome sequencing of selected Philippine mango species is here. And we already have molecular markers for red blotch streak peel and resistance to anthracnose. We have developed hybridity testing SSRs, and also we have data for genotyping by sequencing, and of course the genome sequence of three Philippine mango species. So the outcome that we are working on is to contribute to increased yield from 5.2 tons per hectare to 15 tons per hectare, and to reduce post-harvest losses from 40 to 14%. So this is the mango molecular breeding program that we are trying to push for. We have the first part, of course, is phenotyping. We followed it by genotyping by sequencing using diversity arrays technology. Then we went into mango genome assembly and annotation. And for now, we want to look at the mango breeding applications. So what are the inherent challenges in mango breeding? So the physiology of mango itself is a challenge because, first of all, mangoes have small flowers. They have sticky pollen. The mango is highly outcrossed, but the timing of flowering is difficult. And lastly, most Carabao mangoes are polyembryonic. So to meet this challenge, we can utilize the available germplasm that can be found in the gene banks. We can avail of genomic tools or develop new molecular markers. And it's also very important to understand the flowering habit of mango. So genomic tools can be used or have been used to face some challenges in mango breeding. So we have SSR markers that we can use for hybridity testing. We can also use this for genetic relationships and also for barcoding. Data from genotyping by sequencing can be used to generate or have been used to generate single nucleotide polymorphisms, again, genetic relationships, and to develop markers for important traits. So this project, which was on the full genome sequencing of 3 Philippine mango species, had a general objective to assemble the whole genome sequence of 3 mango species, specifically Mangifera indica, Mangifera altissima, and Mangifera odorata. So we have here their local names, no, Sweet Elena, Pahutan or Paho, and Wani. So assembly would be done through next generation sequencing and to annotate the SNPs produced from the genotype by sequencing data. So the reason why we chose these 3 species is that Mangifera indica, obviously this is the variety that we are most famous for, and it is polyembryonic. It is primarily yellow in color. Okay, Mangifera altissima is monoembryonic. It's self-pollinated. And then Mangifera odorata has some traits related to disease and insect resistance. So odorata is or Wani, is a cross between Mangifera indica and Mangifera foetida. So the specific objectives would be, of course, to sequence and assemble the whole genome of 3 Mangifera species, to annotate the 15,000 SNPs that we got from the GBS data. We wanted also to develop a bioinformatics pipeline suitable for the complexity of the mango genome, to create a public library for the annotated genome of mango, and lastly, to improve and enhance individual and institutional capacity for knowledge and skills in whole genome assembly, bioinformatics, genomic data analysis, and functional genomics. Okay, so for the genome assembly, this is our work plan or our sequence of events. Okay, so we divided the workflow into 3. So the first one is the physical sequencing part, and the second one is the genome assembly part. Okay, and then the third one is the quality test and comparison of our genome with the known reference genomes that have already been published. And then from the last part, we also work on the annotation of the genes or the sequences that came out. So we check for the quality, and then we used Maker, which can which works on gene prediction. So it aligns express sequence tags and proteins to a genome. It produces gene prediction algorithms and automatically synthesizes data into gene annotations, having evidence-based quality values. So for the gene models, now we have the gene models, we subjected them to BLAST-P and to the InterProScan, which provides functional analysis of proteins by classifying them into families and predicting domains and important sites. After the InterProScan, we went into gene ontology, so we made use of merge, go mapping, and GoSlim, and then we have these sequence files coming out from this work. Okay, for the analysis of the quality of our sequences, we took a look at the summary of the assembly size. So for indica, it's 401.51 megabases, which is quite similar to what has been reported for mango. And for odorata, it's quite bigger, while altissima has the biggest size, no, the biggest genome size. And this could be due to the fact that there could be build-up of sequences. Okay, so we also compared how complete our assembly was with Tommy Atkins. Tommy Atkins has its sequence uploaded to NCBI. So we can take a look at the last column. So we just take a look at the percent completed BUSCO. So we have actually Indica has higher rate of completion compared to Tommy Atkins, and this could be because we have less of missing sequences, and then Mangifera altissima has the lowest BUSCO values here. Okay, so in the next slide, we will show just a graph for the summary statistics of the 4 genomes. So there is a high alignment accuracy and mapping rate of the lead pairs, and this actually shows that we are now near complete in terms of genome coverage, and the contigs have high structural accuracy. So this altissima is currently being worked on now, and we are trying to see if we could actually increase the level of completeness for the PAHO variety. Okay, so this is a file showing, or this is a slide showing STAR. These are splice transcripts, alignment to a reference. So here to align our DNA to transcriptome data coming from NCBI. So this data, we took this out from the NCBI, and we can see that the total mapping read rate for Mangifera indica for varieties was 94.74%, but for tissues it was 96.05%. No, and it's quite high when you take a look at this because the transcriptomes, so they're just reference values, and we need more transcriptomes uploaded to the databases. So we only made use of 2, and we have less than 7% that is unmapped because the sequences are too short. Okay, so this is analysis of the SSRs that are present in the genome, and we can see that the predominant motif was AT, and TATA. These are the predominant paired repeat motifs, and there were only a few SSR motifs that were found to be specific for indica, odorata, and altissima, and you can see them here. So this is the interproscan results. They were in this program compares our genomic data with all proteins that are present in the database of the Interpro. Okay, so we have here the green one will show us which sequences are with Interproscan, and then these ones will show us which ones have gene ontologies that are uploaded. And this is the functional annotation for, so we have here, this also comes from the Interproscan. No? So we have here biological processes, cellular components, and molecular function. So for the molecular function, we can see that the highest would be for transferases activity, hydrolase activity, then catalytic activity, okay? And for cellular components, we have the highest for regulation of transcription, which is actually, I think this is logical. And then, of course, for the cellular components, we have nucleus with the highest, followed by plasma membrane. Okay, so this is a listing of the biological processes by specific categories. Okay, and again, this is another which shows us the functional annotation and the family of proteins that they belong to. Okay, so in conclusion, the results demonstrated that highly contiguous phase and complete genome assemblies of the 3 Philippine mango species were demonstrated for structural and functional annotation of gene units, especially those with economic importance. So looking forward, and now that we are done with annotation, and this is going to be followed by uploading our sequences to the Mango database site and also with NCBI. So we're still choosing between the two. Now would come the mango breeding application. So initially, we have already certain traits that we would like to work on. Specifically, our group is interested on working with disease resistance genes. So we have in the pipeline a project proposal on transcriptomics of mango for anthracnose and sessid fly resistance. Okay, so that's one. And then in the works, we have the full omics for mango. And another aspect that we would like to work in is to make use of the current SNP markers that we have for screening in hybrid population. So the hybrid population still have to come, but hopefully in the very, very near future. And with that, I end my talk, and I'm ready to answer questions. Thank you very much.
Thank you so much, Dr. Weng. Through Dr. Weng's talk, we are able to see that biotechnology is not solely focused on producing technologies. Rather, we can see that it is also a discipline, a science that seeks to pave the way for more studies to bear fruit. Dr. Weng and her team continue to pave such ways so that our researchers can use new data to the advantage of their own endeavors. Once again, thank you po, Dr. Weng. Once more, we would like to encourage everyone to prepare their questions as we are going to have another open forum after our last speaker. That said, let's go ahead to our last speaker of the day. To introduce our last speaker, he is a university researcher under the Genetics Laboratory, Institute of Plant Breeding, University of the Philippines, Los Baños, or IPB-UPLB. He works on plant biotechnology, mainly on genetic engineering for introducing novel traits, as well as the elucidation of gene function. He also works on the mutagenesis of genes related to fruit browning, insect defense, parthenocarpy, and doubled haploid induction of eggplant via CRISPR-Cas9. He earned his MS in genetics and BS in agricultural biotechnology degrees in UPLB. Everyone, please give our last speaker of the day a round of applause, introducing Mr. Mark Gabriel S. Sagarbarria for his talk on the utilization of new breeding techniques, or NBTs, for eggplant improvement. Good afternoon, sir. How are you doing this afternoon?
Hello, good afternoon. I'm doing good, and I'm glad to be here.
I'm thrilled to hear that, sir. Before you start, can you please tell us what NBTs mean as a concept in general?
Okay, so NBTs or new breeding techniques are a group of biotech tools that can be used for plant breeding. So they are emphasized as new because they can help develop new plant varieties faster compared to conventional techniques. So my talk later will focus on one type of NBT, which is known as genome editing.
Thank you for that answer, Sir Mark. Now, I hope everyone is ready. Sir Mark, please take the virtual stage.
I am Mark Gabriel Sagarbaria, and I am a university researcher and also currently the leader of the New Breeding Techniques or the NBTs group at the Genetics Laboratory, Institute of Plant Breeding in UP Los Baños. So my talk today will be about the application of CRISPR-Cas genome editing in eggplant, and this will focus on the highlights of our study in the DOST-PCAARRD project and entitled Development of Improved Eggplant Varieties with New Plant Defense Genes for Multiple Insect Resistance using Innovative Technologies under the leadership of Dr. Lourdes Tailo. So here is an overview of my presentation. First, I'll be giving a short introduction on genome editing, specifically the CRISPR-Cas9 technology, then how it is applied in plants, and some of the requirements that you need before you start applying this technology. But mainly, I'll be talking about how we applied it in eggplant, so how we started, what we have done, and where we are now. Then, the next steps that we need to take for eggplant genome editing. And lastly, I'll bring up some challenges in the adoption of genome editing here in the Philippines. Okay, so genome editing involves a group of technologies that give scientists the ability to change an organism's DNA, and these include meganucleases, zinc finger nucleases, the TALENs, and also the CRISPR-Cas system, which is the most popular because it is simpler and more feasible. So all of these technologies have the capability of inducing a double-stranded break in the DNA, and the cell needs to repair the double-stranded break, and this may result in insertions or deletions that may disrupt gene function, and in the presence of a template, precise DNA editing can be done, or gene insertion. Genome editing is a tool classified under the New Breeding Techniques, or the NBTs, which is a suite of modern biotech tools that facilitate the development of new plant varieties with desired traits faster than conventional plant breeding techniques. And different traits can be produced depending on the type of NBT that you use. So this is a list of the available NBTs, and genome editing technologies utilize site-directed nucleases, or SDNs, or oligonucleotide-directed mutagenesis, or ODM. So if you want to learn more about the different types of NBTs, you can refer to this review by Ordonio et al. If you're interested to know more about NBTs. The CRISPR-Cas gene editing system has two major components: the Cas, or the CRISPR-associated protein, which is an endonuclease that can cleave the bonds linking nucleotides in the DNA, acting as the molecular scissors that results in the double-stranded break. But to do that, it must first recognize a sequence known as the protospacer adjacent motif, or the PAM, for cleavage to occur. So a commonly used Cas protein is Cas9, which recognizes the 5' NGG 3' sequence. So next we have the single guide RNA, or the sgRNA, and this consists of a 20 nucleotide guide sequence known as the CRISPR RNA that is engineered to an RNA scaffold known as the tracer RNA. So the sgRNA directs Cas9 to the target sequence, which is determined by, the CRISPR RNA. So when these two components are delivered or are expressed in the cell, they combine to form the Cas9 sgRNA ribonucleoprotein. This seeks out the target sequence within the cell's nucleus, and if there is sufficient complementation of the CRISPR RNA to the target, and there is a PAM sequence to be recognized, Cas9 will cleave the phosphodiester bonds in the DNA around 3 bases upstream of the PAM site in both strands, and this results in the double-stranded break. Researchers and breeders have used the technology to perform single knockouts to produce valuable novel traits in crops, and they have applied it to elucidate gene function and regulation in plants. Examples for these include nicotine-free non-transgenic tobacco and identifying resistance genes for plant pathogens. There are many more applications, and even epigenetic changes are possible, as well as chromosome restructuring. This is an example of a common workflow for plant genome editing. But before you start, you must first identify your target sequence in your crop of interest. And when you do that, you have to design your single guide RNA. So specifically, in your target sequence, you are looking for PAM sequences and identifying which CRISPR RNA will be the best to edit that gene. So after that, you have to clone your Cas9 and sgRNA sequences into an expression vector, and then deliver that vector into your crop of interest. And lastly, you will be screening your transformed cells or plants for mutations in your target sequence. Based on that workflow, we can come up with a list of requirements for plant genome editing. So first, you need knowledge of your target sequence. Second, we need a reference genome to identify any possible off-targets. Third, we need a delivery system, which is usually agrobacterium-mediated transformation. And last, we need a regeneration system for transformed plant cells. So let's check how many of these requirements we had for eggplant when we first started. So actually, we only had the reference genome, which was already publicly available from Soul Genomics. So at this point, when we first started, we still had a lot left to do. So we decided first on our target genes that we wanted to edit, and to demonstrate genome editing, we chose to edit the polyphenol oxidase, or the PPO genes, in eggplant, and these genes code for enzymes that catalyze the oxidation of phenolic compounds, and this results in the flesh browning when we cut our eggplant. So knocking out these genes has shown to reduce browning in CRISPR-edited mushroom, and knocking out the same genes for eggplant probably would also lead to reduced browning, which is a potential consumer-oriented trait. But aside from that, literature also suggests that PPOs have a possible role in plant defense, which could lead to studies on PPO gene function in knockout mutants. We performed PPO gene sequencing and sgRNA design as one of our first activities. So using the available reference genome, we designed primers and sequenced our target genes, which are PPO4, PPO5, and PPO6 in our target eggplant genotype. So now, we've checked off one of the requirements in our checklist, which is knowledge of our target sequences. And because of this, we were able to design the sgRNAs to target these 3 genes at different regions indicated by the red arrows. So each arrow indicates the location of one sgRNA, and because of the sequence similarity between these 3 PPO genes, each sgRNA can target 2 to 3 PPO genes. The project commenced in 2018, and one of the biggest hurdles we knew coming into this was the regeneration of eggplant. So we first established a regeneration system without transformation from 2018 to 2021. So this took us quite a long time to develop or optimize, but we were able to get a 30% regeneration rate. So this figure shows a summary of that process. So from eggplant cotyledons sown in vitro, we were able to regenerate a whole plant. So after that, we proceeded with agrobacterium-mediated transformation by late 2021, and we were able to obtain transformed regenerants, or the T0 generation, by late 2022. However, we were only able to obtain around a 4% regeneration efficiency in transformed plants. So now, after around 4 years, we were able to develop a transformation or delivery system for the CRISPR-Cas components and the subsequent regeneration system. So in these types of experiments for transformation and regeneration, you'd be happy to just obtain one acclimatized and survived plant in the greenhouse. But in our case, we were able to acclimatize 9 plants which have survived and flowered. And this figure shows the summary of the whole process of transformation and regeneration. And we show that the plants produced fruit and seeds. And here we are with PCAARRD project monitors with the first batch of the T0 plants. Based on our screening methods, we observed that 2 out of the 9 plants had edits for our target genes. However, one of the 2 plants with the edits had died due to Phomopsis disease. But thankfully, we were able to produce the next generation, or the T1, through self-pollination of the T0 plants. So here we are harvesting the physiologically mature fruit and harvesting the seeds. We had sequenced the surviving plant for our target genes, and we observed simultaneous editing of the 3 PPO genes, PPO4, PPO5, and PPO6. And for reference, the unedited or the wild type sequence are indicated as the reference sequence. And the edited samples with mutations or the edits are below. So upon checking their amino acid sequences, we observe early stop codons or frame shift mutations for each type of mutation, signified by these asterisks. Aside from that, we also observe 2 types of mutations in PPO4, indicating that there is some mosaicism that is present in the T0 plants. So because of that observation, we inferred that the regenerated plants are mosaics. So what does this mean? So in the context of the CRISPR-Cas gene editing system, this implies that the plant contains the CRISPR-Cas components, but the edits are different for each cell, which can be due to varying Cas9 activity. So this is because the activity of Cas9 may be different in each cell as the plant develops. So for example, let us assume that during transformation, the CRISPR-Cas components are successfully integrated into the plant genome. However, it is possible that Cas9 will not be active before the plant cell divides. So this means that there may be different mutations that occur as the plant regenerates, and mutations that occur in the flower or the germline tissue will be inherited. Despite the plants being mosaics, we observe that editing efficiency increases over the generations. So firstly, the T0 sequences that I showed earlier were obtained from leaf tissue, and it is possible that these were not inherited in the T1 generation. So for the T1 generation, we estimated the editing efficiencies of our target genes using Sanger sequencing, and we were able to obtain around a 60 to 80% editing efficiency in the T1. So meaning that mosaics are still present. A similar study by Mawili et al. In 2020 that also edited eggplant PPO genes observed similar T1 editing efficiencies of around 50 to 60%, but their T2 editing efficiencies were drastically increased to 80 to 100%. So this means that generation advancement can be done to increase editing efficiencies as long as Cas9 is still present and active. And generation advancement increases the likelihood that mutations occur in the germline tissues or the flowers that will result in homozygous edited plants. To summarize, our results so far have successfully demonstrated a working genome editing platform for eggplant. So since we have a working protocol, it will be easier to target other genes, which will be soon to follow. But for the PPO edited lines that I discussed, we must still continue generation advancement to obtain homozygous mutant lines. Afterwards, backcrossing should be done to remove the transgenes, which is the Cas9 gene itself. And after doing so, after obtaining the homozygous lines that are transgene free, the fruit traits must be assessed to determine if fruit browning and PPO activity is reduced. Lastly, I want to bring up some challenges in the adoption of genome editing technologies in the Philippines. So first, there are some heavy infrastructure requirements as tissue and cell culture capabilities are required. Aside from that, the facility must also be approved for biosafety, and the laboratory must also be able to perform molecular cloning techniques. And of course, we need experts to perform the lab work. So the bottom line is you need infrastructure, resources, expertise, and a lot of time. But despite these challenges, I want to encourage researchers to pursue the application of NBTs like genome editing in basic research and crop improvement, as from our experience here at IPB, we were able to establish a genome editing platform for eggplant within 5 years. So it took longer than expected because we had trouble with regeneration, but future applications of genome editing in eggplant will be made much easier since the platform is already available. And the same will also be true for other crops once the requirements for NBTs are met. So I'd like to end this presentation by sharing with you all news from a few months ago that a gene-edited banana developed by a UK company has already been approved for importation and propagation here in our country, and it has been determined as non-GMO. So we should be strengthening our efforts in applying these technologies to improve Philippine crops, especially since the regulatory framework for the approval of crops subjected to NBTs like genome editing is already available. Thank you very much for listening to my presentation, and I look forward to entertaining comments and questions from the audience.
Maraming salamat po, Sir Mark. Plant breeding is one such concept that a lot of people know has unpredictable results while also taking a lot of time. Through the works of researchers like Sir Mark, we can, in more ways than one, improve on the techniques that will not only shorten the time needed, to see results but also provide us with overwhelmingly positive returns when it comes to plant breeding. Once again, maraming salamat po, Sir Mark. With that, all of our speakers have taken to the virtual stage and shared their works with our live audience and with all who are interested. To our audience, you may type your questions in the comment section of our FB live stream. Once more, I would like to call on both of our recent speakers to the stage, Doc Weng and Sir Mark. Magandang hapon po sa inyo. Ready na po ba tayo sagutin ang questions ng ating live audience?
Ready.
Ready na po.
Ayan, mukhang ready na po si Sir Mark at si Doc Weng. Now to start our question and answer portion. This is from Charlotte Collado of Baybay City, Leyte. What were the difficulties encountered when you worked on the mango genome? This question is directed to Doc Weng.
Okay, so there's not actually a lot of difficulties. We just had to, of course, it was a project, so meron siya a problem, with procurement or with administrative work. But for the work on molecular biology was just getting enough quality mango DNA, and then we sent out samples for sequencing. And when it came back, we had to do the bioinformatics. And we didn't have training when we started, but we got it during the time that by the time that the sequences arrived. We had more of a problem with the internet connectivity because most of the software that we used were freeware, so we don't have very fast internet. That was what we had. But all the others, it just took time because of slow internet connection. But otherwise, it's not true. So we can do it for all other food varieties. Yes, yes.
Thank you for that answer, Doc Weng. Next question, please. So this is from Alvin Ventura of Ligao, Albay. What is different from the established eggplant genome editing platform from other published genome editing protocols in other crops? This is addressed to Sir Mark.
Okay, so the major processes are the same. But what is different is that we use the single guide RNA that is specific for the genes found in eggplant. So aside from that, the tissue culture and regeneration that we performed is highly genotype dependent. So we used a Philippine genotype in our regeneration and transformation. So if you will be using a different eggplant genotype, the results may vary, so you may have to do additional troubleshooting or optimization.
Thank you so much for that answer, Sir Mark. Another question, please. This is from Enrique Portajada to Dr. Ocampo. Can you provide us a glimpse on how costly whole genome sequencing, or WGS, project is in the Philippines? How can researchers entice funding agencies to fund their WGS project? Thank you.
Okay, so two questions. How costly was genome sequencing? So when we started with project proposal, we asked for, I think, 13 or 14 million, but 5 million of that went into the server to hold that information. So we separated it, and then we had 3 million for 3 years, and we got 7 million for 3 years, and we got 3 sequences. So the whole genome sequencing cost has actually been decreasing because of different platforms that are present. So when we started, we were only looking at one genome of Mangifera indica, but later we added two because the prices really went down. So I think it's not really that expensive. We had one project assistant and one full-time researcher, myself, and then also we had two consultants or project staff. So it's not really that expensive. And then the second one is that genomic data is really very important. All these applications that we can do now, like for example, in genome, they come from genomic data. So you not get the genomic data or the whole the whole genome profile if you don't do the whole genome sequencing. Funding agencies are willing to fund genome sequencing if the plant or the crop is really important. Like how we are doing sequencing for millets, and also for medicinal plants. So there's a lot of work that's going. Because we need this data to move forward with our crop improvement, with utilization of the protein product. So all these are based on the genome. So you should not be discouraged, Mr. Enrique. Okay, thank you.
Thank you for that answer, Doc Weng. Now proceeding to our next question, please. This is from Lin B. Resurrection. To Sir Mark, can the technology be used also against Asian fruit borer like the one used in the GM eggplant? Sir Mark?
Technically or theoretically, you could find a gene that will be linked to the susceptibility of Asian fruit borer, or sorry, eggplant fruit borer. But the problem is that it's hard to find a gene that is linked to the susceptibility of a herbivore like the eggplant fruit and shoot borer, and then knock it out, so that if you knock out that gene, the eggplant will no longer be susceptible. So that's why the GM eggplant, BT eggplant, incorporated a gene that will confer the resistance to eggplant fruit and shoot borer. So theoretically, it's possible, but you would have to find a gene that is linked to the susceptibility of the eggplant to the pest, which is difficult to do, or it may not also exist.
Thank you for that answer, Sir Mark. Now proceeding to our next question, please. Is from John Carlos Suarez of Samal Davao City. What follow-up work is now being planned? And this question is directed to Doc Weng.
Okay. So thank you. I wanted that question so that I can plug in our proposal. So now we have a proposal for the transcriptomic analysis of mango to check for the resistance and then anthracnose and recall is excessive fly. Also, currently, as of this moment, we are waiting for our slot in the Director's Council. So one of my students, previous students, will be presenting this proposal. And another one is another proposal that we are making is for metabolomics of mango plants, parts of the plants and the fruit. And of course, the next, which is for me very important, is to go into breeding of mangoes. Because if you look at the Philippines, we really do not breed for new varieties. We just select and then collect and then we propagate. But we do not have large-scale efforts for breeding. And I think the use of genomic data will be very important here in order to select for mangoes with good traits, hybrids that have the taste that we 2031 because mango takes before you can see the fruit of your labor. But with markers that are derived from technology, you can hasten the selection process. And for now, that's what we are thinking of. But there's really a lot that so we are sharing also sharing our data. You can just download it once you upload it to the databases, and then you can use this for your work. Yes,
Thank you for that answer, Doc Wen. Now proceeding to our next question, please. This is from Lucas Franco of Quezon City. If a research group is interested in using genome editing for plant improvement, what should be their priority? And I think this question is directed to Sir Mark.
That depends on the crop that you want to edit or improve. But generally, one of your top priorities should be the tissue culture protocol or facilities. But let's say, for example, you're working on a crop wherein you can transform the flower tissues directly. So that can be the case for rice. It's a technique known as floral dip, wherein you can directly transform the flowers. So by using that method, you can sort of bypass the tissue culture requirement. So, but you need a facility that can be approved for biosafety. And so, ayan, that should be your next priority, yung biosafety facility.
Thank you for that answer, Sir Mark. Now proceeding to our next question. So this is from Angelo Atienza from Mati, Davao Oriental. What are the potential uses of the mango genomes that your team has produced? This is for Doc Weng.
Yes, currently you can actually, directly, you are screening for a certain trait. You already have gene polymorphism SNPs, that you can screen, that when is first screen. So that is the direct effect. So that is what we have. Then we also have developed assessor markers for hybridity testing because most of the mangoes that we, for example, if you're crossing mangoes, then if you are using polyembryonic varieties, means that one seed is going to produce several seedlings. And one of the seedlings will be the hybrid, another of the seedlings will be the mother plant. So before you grow them out, you can actually use all markers to identify which one is the hybrid and which one is the clone of the mother. That's for the of the mango. And also, you can look for gene sequences and scan it for sequences that you can use for breeding program. And also, you can take a look at the metabolites and you can do it through gene expression analysis. So, sa ngayon po, this is for study, but if you come up with kits for identifying, for example, Carabao, which one is Indian mango, which one is another variety of mango, then that will have hopefully to be in the near future. We can actually develop that. For now, we have all these
Thank you po, Doc Weng. And I'm being told that that would be the last question for our open forum. Maraming salamat po sa inyong pagsagot, Doc Weng and Sir Mark. Sigurado na maraming natutunan ang ating live audience mula sa ating open forum. With that, both of our open forums have ended. If any of our viewers today have further questions, you can reach us through our email at crd@picard.dost.gov.ph. You can see the same email address flashing on your screens. Again, we are grateful to have witnessed the best and brightest of Philippine biotechnology on our stage today. Of course, with this, we would also like to formally acknowledge their presence and willingness to be with us today. Before we proceed to the end of this webinar, we would like to award these certificates for the efforts and expertise we've witnessed for the past hours from our last two speakers. Please allow me to read the citation once more. The Department of Science and Technology, Philippine Council for Agriculture, Aquatic, and Natural Resources Research and Development, DOST-PCAARRD, and the University of the Philippines, Los Baños, present the Certificate of Appreciation to Dr. Eureka Teresa Ocampo for serving as a resource person for the webinar titled SNT Agri-Aqua Biotech Applications, held on November 21, 2023, 2 p.m. via StreamYard and Facebook Live, given this 21st day of November 2023. Signed by Dr. Reynaldo V. Ebora, Executive Director of DOST-PCAARRD, and Dr. Reniel S. Pamplona, Project Leader from UPLB. Last but certainly not least, we would like to present the same Certificate of Appreciation to Mr. Mark Gabriel S. Sagarbaria. Again, thank you very much po for sharing your knowledge with us this afternoon. Maraming salamat po sa inyo pong dalawa. To our audience, please give our speakers a round of applause. Maraming salamat po sa inyong lahat. This webinar wouldn't have been remotely possible without your time, efforts, and undeniable expertise. Now, to formally end our webinar, I would like to call on our very own Deputy Executive Director for Research and Development, Dr. Juanito T. Batalon.
Good afternoon, everyone. I would like to thank you all for joining us in an engaging discussion in today's webinar on SNT Agri-Aqua Biotech Applications as part of the 19th National Biotechnology Week celebration. The technologies presented by our resource speakers this afternoon are among the front runners in advancing the application of biotechnology in the field of agriculture and aquatic resources. We are thankful for the hard work, continuous support, and unending passion of the project teams in delivering the expected outputs of their research. In particular, we are thankful for the guidance and leadership of the experts from the National Fisheries Research and Development Institute and the University of the Philippines, Los Baños. Our experts not only gave us a good discussion on the accomplishment of the projects, but also help us better understand and appreciate biotechnology and its application in the agriculture, aquatic, and natural resources sector. Rest assured that the DOST-PCAARRD will continue supporting the SNT development in the ANR sector, utilizing biotechnology to improve agricultural and aquatic production while safeguarding crops, livestock, and fisheries against production challenges. We hope to showcase more significant outputs of these programs and projects in the coming years, not just in NBW, but in other platforms too. On a final note, I would like to thank everyone for attending this webinar, and I hope that you found this activity useful and engaging. Likewise, I hope that you will continue joining the week-long celebration of the 19th National Biotechnology Week. Make sure to like and follow the NBW and DOST-PCAARRD page on Facebook to learn more about the exciting activities and webinars about the latest biotechnology in the country. Once again, magandang hapon at mabuhay tayong lahat.
So much, Dr. Juanito T. Batalon. Nais din po namin magpasalamat sa mga live viewers natin na sumabaybay sa amin sa hapong ito. Sana ay marami kayong natutunan at patuloy nyo po sanang ibahagi din ang mga narinig ninyo sa inyo pong pamilya, kaibigan at iba pang kakilala. Maaari naman kayong makakuha ng e-certificate sa pamamagitan ng pagsagot sa aming CSF or Customer Satisfaction Feedback Form na matatagpuan sa link na nakalagay sa screen at sa ating comment section dito lang po. Piliin ang webinar on SNT Agri-Aqua Biotech Applications sa ilalim na activity title, sagutan ng form at ipapadala namin sa inyo ang inyong e-certificate sa loob lamang ng 1 linggo. Again, I am Ross Pinido, and that was the 19th National Biotechnology Week's webinar on SNT Agri-Aqua Biotech Applications. Maraming salamat po sa pagdalo.